Good evening, everyone. Let's start the session with the Omkar. Heartfelt welcome to all the participants of the NMO Med-Katalyst Foundation. Today we have a biochemistry session. The faculty member is Dr. Vijaylakshmi Madam, Professor of Biochemistry in GSL Rajahmundry. Today our topic is molecular biology. But today we have an interactive session. You can ask your doubts. It is a privilege to have Dr. Vijaylakshmi Madam with us today. We sincerely thank you for graciously accepting our invitation, Madam. With great respect, I now request Dr. Vijaylakshmi Madam to kindly take over the session and guide us with your valuable insights.
Thank you very much. And so nice of this platform, which is NMO platform, so that it helps to educate, and it has so many other activities. And I know that they're doing wonderful work here. So what I thought is, like I've seen last time, there was hardly any interaction. So I was wondering, if you all ask a question related to molecular biology, then I can go ahead and say, okay, this is how you're supposed to answer. That is how it's going to be better. So can anyone tell me, raise your hands and we can unmute you, and you just tell us that this is what you want, or you can put it in the chat box so that we get an idea of what is it that you want in molecular biology, which question, how to write. Then I'll show you the slides. I'll also explain to you what is very important here, how you're supposed to write that answer. If you get it as a 5 marks question, if you get it as an essay question, it will be useful. So can any of you all give their inputs in the form of a chat, or you can unmute yourself. If you raise your hand, we'll unmute you. And yeah, I can see Hussein. Then I can see Varshini, Sanjana, Lokesh, Kartikeya, Swarupa. So can any of you all give your inputs? You can put it in the chat. Because if it is just for listening to that, you have so many videos which you can just go through those videos. But if you want that it has to be in the form of a question answer session, then it is good that you all ask those questions. These are the questions. Yes, I think I have got one chat received. What was the question? Man, can you explain post-translational modifications? Wonderful. So what we'll do is I'll just show you that, because post-translational, post-transcriptional, both are important, right? So we'll just look at post-translational modifications like how you have said. Okay, so we'll go to translation. And so first thing that I want you all to understand is, see the name, many of you all will get confused between post-translational and post-transcriptional. That is the common confusion which you all have. So remember that post-transcriptional is only after transcription. So I think you all know, no? Transcription, where DNA is going to become RNA. That is transcription. What is translation? RNA is going to become a protein. That is translation. Now they are asking you a question on post-translational modification. So when they're asking you that, so please see that you read the question twice, 3 times, so that you know it is post-translational. And when you know that it is post-translational modifications, then you can go ahead and then you can answer that. So let me just share my screen, and then we will look at this post-translational modifications. Yes, I hope you can see the screen now. And I'll just tell you about this post-translational modifications. Right. So here you can see that what is the use of having a post-translation modification. So when you get a question like this, you explain that This is, you have a native protein being formed. What do we mean by a native protein? An RNA has got translated into a polypeptide chain. Okay, so this polypeptide chain, now it is just a straight chain. For it to become a tertiary structure, it should undergo some changes, because we know that in the structure of proteins, it is only the tertiary structure which has complete biological activity, right? So to form the tertiary structure, what all should happen? So the uses of this post-translation, after it has got translated from the RNA and it has become a polypeptide, what are the modifications which have to occur? One, why is it there? Because it will cause stability of the protein, right? Whenever you have a tertiary structure, or supposing there are more polypeptide chains for that protein, then you will get a quaternary structure. Then the bonds will be different. And these are the bonds which are going to make it stable. So first, the utility of a post-translational modification is to cause stability. Second, biochemical activity may increase. Like we know that whenever we have collagen, what is that post-translational modification? It can get hydroxylated. At which amino acids? You can have either proline or lysine. So when they get hydroxylated, and then the stronger collagen molecule is going to be formed. So for biochemical activity, and then only it is more mature to do its function. Third, protein targeting. Protein targeting is, now they should have this polypeptide which is formed, a signal peptide should be attached to it. What is the function of that signal peptide? To carry it to that particular subcellular compartment. So that is called protein targeting. So you have a subcellular compartment like a peroxisome, where some enzymes have to go there. Now, supposing the signal peptide is not there, then you get a disease which are called peroxisome biogenesis disorders, one of which is Zellweger syndrome. Okay, so that is another. For protein signaling, so we know that for protein signaling, you have a cascade phenomenon. You must have read about this in glycogenesis, glycogenolysis. We have seen that how it is that you are having a receptor for some hormones like a glucagon. You have a receptor for epinephrine. Now, glycogenolysis has to take place. So they will sit on that particular receptor, these hormones, and they will activate second messengers. Now, these second messengers are going to cause a lot of phosphorylations. Now, this is nothing but a signaling cascade which has happened. So for a protein, if it gets phosphorylated, then some proteins become more active, some proteins become less active. What I mean to say is enzymes or proteins. Okay. Now, what are these post-translational modifications? So for that, you should know that some of the post-translational modifications can also be, as it is being synthesized, it might get translated. That's why it is also called as co. But remember it as post-translational modification. One example for that is trimming. Trimming is what? Proteolytic degradation. So when you are having a proteolytic degradation, what is actually happening to it? We know that we have insulin in the form of pre-pro-insulin, which is a very long polypeptide. Now this is finally going to get cut and become finally how much? It is going to form an insulin which is having about 51 amino acids. So that is why this is called trimming. Initially it was not active, later on it becomes active. Okay, similarly, you have your digestive enzymes, right? Chymotrypsinogen, trypsinogen. So you know that these were actually not active. But when a small peptide has been removed from these proenzymes, then they become active. So that is another example. Next, inteins. I think inteins, even if you don't write, it is okay. So just ignore inteins. This is not so important. This is usually important only if you want to get very high marks. But otherwise, generally, you remember trimming is one. Next, remember this covalent modification. This is important. Covalent modification is important because it is commonly taking place. You have phosphorylations. Phosphorylation, addition of a phosphate group. You remember your enzymes like glycogen synthase, glycogen phosphorylase. For phosphorylase, a phosphate group should be added. Then it becomes more active. For a glycogen synthase, phosphate should be removed. Then it becomes active. If it is phosphorylated, it becomes inactive. That is how the regulation takes place. Glycosylation, you know, most of your proteins in the body, they are always having some oligosaccharide attached to them. That is called glycosylation. Hydroxylation, we spoke of proline and lysine in collagen. These are the amino acids which can get hydroxylated. Carboxylation. Now, this is very important. If you remember, in clotting factors, we have gamma carboxylation of 2, 7, 9, and 10. So these, they can become activated. Acetylation, an acetyl group can be added. An acetyl group, if it is added, histones are there. When acetyl group is added, now these histones will stop getting attached to DNA. So when they do not get attached to DNA, DNA is now free to undergo transcription or translation. Farnesylation. Now this is seen in very few proteins. This is actually a farnesyl group. If you remember your synthesis of cholesterol, we see that you have a farnesyl group there. So this is actually a lipid part. So lipid part which is being attached to some protein. Then we have the next one is protein folding. Now this protein folding, we know that a protein, when it is formed, you have a primary structure. Then you have some more bonds, that is the hydrogen bonds, which make up the secondary structure. And then we have actually the protein will fold to form the tertiary structure. Now this folding can happen spontaneously, or it will require the help of certain other proteins which are called chaperone proteins. Okay, so this is, and you have to show these examples. So chaperone proteins, you just say what are our chaperone proteins. So these are what it is clearly mentioned, that they bind reversibly to hydrophobic regions of the proteins, and they help in normal folding of a protein, so that you will have the correct biological activity. Only then it will have its correct biological activity. Then you can give examples of chaperone proteins like heat shock protein 70, chaperonin system. Just write these two parts. Okay. So this is one example. Then you have seen the other one. Supposing a polypeptide is having more than one polypeptide, that is a protein is having more than one polypeptide, then what will happen? Those subunits of that particular protein should aggregate. Example is what? You know your enzymes, like you have creatine kinase, which is actually a dimer, or you have lactate dehydrogenase, which is a tetramer, or you have hemoglobin, again you have a tetramer. So they have to now assemble, forming bonds amongst themselves. Then this is another form of post-translational modification. So this is what you have to write, and you can give examples where post-translational modifications are imperfect. Then what will it cause? Then you have to give this example. Protein misfolding disorders can happen. One of the examples for that is cystic fibrosis. So in cystic fibrosis, what is happening? You have that protein which is called CFTR, cystic fibrosis transmembrane regulator protein. So if this protein is functioning well, then you will not have this disease. The chloride channels will be working effectively. Otherwise, it does not work effectively, and all the secretions in the person will become thick. So you have salivary secretions are thick, pancreatic secretions are thick, and they might affect the functioning of pancreas. So that is because of a misfolding disorder in the protein. The other example you can write is prions. So you know prions are what? Proteinaceous infectious particles. So if normally we think what a bacterium can be the causative infective agent, you have a viral infective agent. But imagine proteins themselves are becoming infective agents and they are going to cause disease. So this is because these are the proteins which have not folded correctly. That is why they have become infectious particles and they cause diseases like Alzheimer's disease or Creutzfeldt-Jacobs disease. So if you write this much, this is enough for a question on post-translational modification. And if this is going to come, it usually comes as a 5 marks question, right? So I hope this answers your doubts. And do you have any other? And I hope it has answered your doubt, or do you have some more questions where you would like to again ask some more? Can you just tell me what else you have? Yeah. Okay, Hussein. Anybody else who has?
They asked PCR.
Pardon, PCR? So there is another person who has asked about PCR. So well, let's go to that and see. So PCR stands for
Another question on transcription, ma'am.
Oh, there is a
Can
question.
you explain transcription in eukaryotes and post-transcription modification?
Okay. Transcription in eukaryotes and post-transcriptional modifications. Okay, then we'll take that up now. So first thing is transcription in eukaryotes. It is a little complicated when compared to prokaryotes. So usually the question which they give will be explain in detail about transcription. Right. They will ask you explain in detail about transcription if it comes as an essay, and explain about post-transcriptional modifications. Very rarely they will ask you differences between transcription in prokaryotes and eukaryotes. But anyway, I will tell you about prokaryotes and eukaryotes also. Specifically on eukaryotes, I will tell you all. So we will look at let's go to, right. Okay. So let's just see this. Okay. Right. So I hope you can see this slide, right? So transcription, you know that what is transcription? From DNA, you are going to get RNA. This is transcription. Now, if they give you transcription just generally, write about prokaryotes and eukaryotes also. But if they specifically, and I'm sure you're thorough with prokaryotes, that's why you're asking about eukaryotes. So when they ask you about eukaryotes, what you do is, it is the same thing, initiation, elongation, termination. These first write the definition. After you finish writing the definition, then you divide it into initiation, elongation, termination. Now here the initiation will be different for eukaryotes. What is the reason? You are having histone proteins are attached, right? So first the histones should become separate. This is what you have to write initially. So the histones should be acetylated. When it is acetylated, it is no longer going to get attached to DNA. So it will separate itself from the DNA. Then it is easy for that DNA to have transcription, because now it can act as a template so that you are going to have transcription is possible. Now, what is the difference in initiation? So transcription initiation also same thing like in prokaryotes, it requires a promoter. It has upstream regulatory regions. So I hope you know the meaning of promoter. Now, promoter is the site where actually it is recognized by RNA polymerase. So that is the site where at the +1 of this site, you are going to have actually the start. So it is usually present as a site which is recognized by the RNA polymerase, which goes and gets attached to it. Once it attaches to it, it slowly moves forward till it comes to the transcription initiation site. That is the +1 site. Okay. So you have a promoter. What do you mean by upstream regulatory elements? Now, from the transcription initiation site, which is +1, anything which is going downward, that is after that +1, +2, +3, etc., that is called a downstream regulatory region. Anything before this +1, so which will not be written as minus or anything like that, but anything before the +1, it is called upstream regulatory elements. So transcription initiation requires a promoter. It requires upstream regulatory elements. Now, what are these elements called? They are called cis-acting elements. Now, what do we mean by cis-acting and trans-acting? Cis-acting elements means they are present on that same DNA strand. Okay, that is why they are called cis-acting. Now, trans are, they are produced by some other strand, some other part of the DNA strand they are produced. A particular protein may be produced there, that is after translation. That protein will come and might act here. So that becomes a trans. Okay, I hope this is clear for you. Cis-acting elements are usually bits of the DNA itself. They are bits of the DNA itself, bits of the template strand, which is now acting as a regulator. So that is a cis-acting element. What do we mean by a trans-acting element? It is some other part of the DNA which is actually undergoing transcription, translation, giving you a protein. And that protein is now coming here and acting at some site on this template to help in regulation. then it is called a trans-acting element. Okay, so the cis-acting elements are specific sequences on the DNA template which is going to regulate this. So let us look at this. So now you can see in this, right? So when you look at this, let us look at, this is actually our structural gene, the gene which is supposed to be transcribed. Now you have a box which is called the Tata box. Now this is actually the cis-acting element, which is the promoter site for a eukaryote. It is called the Tata box. It is also called the Goldberg-Hodgkin box. Okay. Whereas in prokaryotes, what did we have? We had a Pribnow box. Here it is called the Goldberg-Hodgkin box. Then we also have another box which is called the cat box. Now this box is around some 70 nucleotides away. Okay, some 70 nucleotides away. And this box which you can see here, this is around, Tata is around -25. That means this area where you can see the exon being written, this is, you can take it as +1. It may be an exon, it may be an intron, but this is the site where actually the transcription is starting. This is the +1 site. Okay. Now these are called the cis-acting elements. So you have a Tata box which is around -25 nucleotides away, cat box which is about -70 nucleotides away. Then you have a GC box. Then you also have some other elements which are called enhancer elements, which are quite further away. So these are all the cis-acting elements which are very close to the gene of interest which is going to be transcribed. Now, so this is just to show you that the Tata box is the promoter here. Some eukaryotic promoters are also there called INR, BPE, etc. They are also other promoters. CAT and the GC are called proximal regulatory elements. Okay, they will decide how frequently this gene has to be transcribed. Should it be transcribed only once? Should it be transcribed more than that? Should it be transcribed 5 times? One example I can give you is, you know about glucokinase, right? So glucokinase is a enzyme, the gene of which will be induced for expression in transcription by insulin. So insulin increases the expression of what? Of glucokinase. So how will it do that? When a person is eating well, then what should happen? Insulin will act. Insulin will increase or enhance the transcription or increase the frequency of transcription of this enzyme or of this gene, which is glucokinase gene. So that is why it will act how? And so you have the CAT box and the GC box will be activated. And these are the proximal regulatory elements which will decide how frequent this transcription should be. Then you also have some enhancers and silencers. Enhancers will enhance the reaction. So these enhancers are actually distal regulatory elements. They act along with the proximal regulatory elements to increase the expression or to decrease the expression. This is to do proximal regulatory are the frequency, how many number of times. Whereas the distal, they are just to either increase the expression, decrease the expression. They will express the gene or they will not express that gene. So these are the elements. Then we come to what exactly, how is it this RNA polymerase comes here? So we all know that we have different RNA polymerases when it comes to eukaryotes. You have RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. We are interested in RNA polymerase 3. Why are we interested in that? Because this is the one which is going to give you your mRNA. That is why we are interested in RNA polymerase 2. Now this RNA polymerase 2, before it can come and act, you can see that you are having some protein here. This is called transcription factor 2D. So these are the proteins. So these are the proteins which are now going to come and they are going to bind to which area? They are going to bind to this Tata box. The Tata box, which is also called the Goldberg-Hodgkin box. So this TF2D, transcription factor 2D, these are also called general initiation factors, they are called. So they are going to come and they will bind to the Tata box. So, and along with other factors. So you need not mention all these other factors also which are there. 2F, 2B, H, E, A, etc. Don't mention because you can't remember that. Just remember this transcription factor 2D, which is going to come and bind to the Tata box. Okay, so the Tata box. So what is this 2D made up of? It is made up of a Tata binding protein and transcription associated factors. If you can remember that, you can write that. Otherwise, you just write factor 2D. Transcription factor 2D will come and it will bind to the Tata box. And when it binds to the Tata box, it calls upon the other transcription factors, which are called the general transcription factors, which will come and bind to this. And these factors will bring the RNA polymerase also to this site. And what is this site? The promoter site. So this is about the complex which is formed in pre-initiation. So then the rest is same. The elongation will be the same. That is, from that +1 side, it will start going on elongating. So what all it will keep adding? This RNA polymerase will keep on adding these ribonucleotide triphosphates will enter as substrate, but the monophosphates are going to get attached, and it is from the 5' to 3' direction it will take place. For prokaryotes, it was transcription translation, simultaneously it will happen. Whereas for eukaryotes, because of the membrane, nuclear membrane, transcription happens separately, translation happens separately. Termination. Now, termination is slightly different here. So here, what do you have? You have a termination signal is there, which is, or a sequence, you can call it. The termination sequence is, this is the sequence. This is present where? It is present on the template strand. This sequence is present. So now the termination sequence is this, and the endonuclease will cleave 15 bases 3' of the sequence cleavage and polyadylation tail. So that if it is AAUAA here, that means this is there on the RNA. So the exact opposite of this should be on the DNA or the template strand, right? So on the template strand, if here it is TA here on the RNA, then you will be having here T, right? So this is how you have. Then here it is U, here you will have A. So this is the termination sequence. That means on the strand of the template strand, you are going to have exactly complementary nucleotides will be there. Now this is the sequence which is formed in the RNA. Now you are going to have, this acts as a signal, and you have the endonuclease coming, and it is going to cleave about 15 bases 3' of the sequence. And you also have the addition of the polyadenylation. That means AMPs are going to get added after that. You can see all the A's here. No? So AMPs are going to get added. Adenosine mononucleotide phosphate is going to get added. So these are all going to get added, and that also acts like a signal. So once it acts like a signal, you are going to have immediately, it is going to be removed from the attachment to the template strand. It will be separated, and after that you will have the post-transcriptional modification. So it was easy for you about prokaryotes. Here the only difference is, remember, in initiation you have some difference. What was the difference in initiation? You have to talk about transcription factor 2D. You have to talk about others, things like the promoter sequence. You have to talk about proximal regulatory elements, distal regulatory elements, Tata box, which is around 25 nucleotides away, that you will be talking about. Elongation is same. Termination, you have to talk about the termination sequence. So this is what, just to show you, this is the signal. It is going to be cleaved, and now you're going to have a polyadenylated mRNA precursor is there. So this precursor, now we will see what is going to happen within the nucleus. So within the nucleus, what is going to happen in this? So what is being produced now is called a hnRNA. That is, heterogeneous nuclear RNA is being produced. This is also called the primary transcript. So this is only there in eukaryotes. So these are very large and large immature forms of mRNA. Okay. So then what are the post-transcriptional modifications? So you can see that you have capping, which is present at 5' end, tailing, which happens at 3' end. There is something called mRNA splicing. Then we have alternate mRNA splicing and RNA editing. These are the 5 types that you have to write. This is for what will you write? It is for only hnRNA, which is going to give you mRNA. So similar thing will happen even for tRNA. There is something different, post-transcriptional modification. For rRNA also, you have a different post-transcriptional modification. You have to remember that. Don't think if they ask you post-transcriptional modification, you have to write this and that is over. This is only for post-transcriptional modification for hnRNA, which will give rise to mRNA. Okay. Right. So now let us look at capping at the 5' end. So at the 5' end, what are you able to see? So you can see this methyl guanosine triphosphate cap. You can see that this entire thing is being added here. What is the use of this? This cap is added because it helps in initiation of translation. Next, it prevents the digestion by 5' exonucleases. It prevents. So that is why a capping is done. Now let us look at the tailing. Tailing is at the 3' end. What is the use of adding so many polyadenylate residues in the tail? Because this tailing is again taking place in the nucleus and it protects against 3' exonuclease activity. There are so many enzymes which are there which can damage this RNA. So that is why it protects against this damage and giving stability. Therefore, it helps in translation. So we have to talk about the capping at the 5' end, tailing at the 3' end. Third one. So if you can draw the diagrams, it is well and good. Otherwise, at least make a flow chart and show it that this is how it happens. Third is splicing. So what do we mean by splicing? So splicing is when you are going to cut the introns and then join the exons together. So just mention that for this, we require some proteins which are rich in uracil. Now, these proteins which are rich in uracil, they are the ones which are going to cut at certain points, that is on that intron. They will remove the intron completely, join the exons. So what will happen if there is a damage, and that is this uracil rich proteins are not there? Then it will cause the introns will continue to be there, and therefore you will get abnormal proteins. One of the example for that is beta chain of hemoglobin. If you have a mutation at the exon intron junction, then the intron cannot be removed and you will have an abnormal protein which can be formed. Now I showed you some uracil rich proteins, right? So those uracil rich proteins are actually ribozymes. Okay. So what are these ribozymes? You know, ribozymes are RNA enzymes. So these are called SNRPs here. What is SNRPs? Small nuclear ribonucleoprotein particles. They are called SNRPs. Now these are the SNRPs which help in cutting the introns, joining the exons. Now supposing you develop antibodies to SNRPs, then you will have a disease which is called SLE or systemic lupus erythematosus. So this is a point where you can write as one of the clinical importance in post-transcriptional modification. Next, we spoke about alternate splicing. In some places, they give it as alternative splicing. So what actually happens in this alternative splicing? So normally what we've seen, introns will be cut, exons will be joined. But in some places, what happens is some exons will be expressed, some exons will not be expressed. One of the example that you can see here is in smooth muscle cell, there you can see different type of exons being joined. Whereas in a striated muscle, again, different type of exons are joined. So what are those exons which have been joined in the smooth muscle? In smooth muscle, you can see 2 is there, 3 is not there, right? Whereas when you look at the striated muscle, 3 is there, but 2 is not there. Same thing you can see here. 11 and 12 is there here, but 14 is there in the smooth muscle. So here, the exons have been joined. So over there, so here alternative splicing is when different exons are being joined for different tissues. This is called alternative splicing. Next, we have RNA editing. What happens in RNA editing? In RNA editing, you have a very large protein. In some tissues, you have a very smaller version of that. How does it happen? The commonest example which is given is ApoB100 and ApoB48. You have heard about these two in lipoproteins. You have heard about an apoprotein. ApoB48, where is it? It is present with chylomicrons. B100 is present with VLDL. How is it that this protein has become so small? Very small quantity, 48%. 48 stands for only 48% of the protein is being expressed. So what is the reason? The reason is for this hnRNA which was there, which is having about 14,000 and odd bases, in that at the 6,000 and odd position, actually it was, the triplet was CAA. Now the CAA editing has happened, from cytosine ammonia has been removed and it becomes uracil. So once it becomes uracil UAA, a stop codon has come now. So therefore the protein is shorter. Therefore it is only 48%. This is called RNA editing, right? So these are the 5 points that you have to write for post-transcriptional modification of mRNA. Now, when we come to transfer RNA, if you can remember, then just say that transfer RNA also undergoes a lot of changes, as you can see at the 5' end, at the 3' end, it undergoes it at the anticodon arm. Then you have some bases can be methylated. Then you are having abnormal bases can be there. You have DHU, dihydrouracil. You have pseudouridine. This mark stands for pseudouridine. The D stands for dihydrouracil. Then you can see here that initially you were having about UU, etc. at the 3' end. Whereas after the post-transcriptional modification, it has become CCA. So these are the changes in tRNA. This is what you can mention. Then the same thing happens in ribosomal RNA or rRNA. So we know that rRNA has so many types. You are going to have a 5.8S RNA, then you're having a 28S ribosomal RNA, 18S ribosomal RNA, 5S ribosomal RNA. Now this 18S, 5.8S and 28S were actually a part of 45S rRNA. So this undergoes splicing again so that you have the formation of separately an 18S rRNA, 5.8S and 28S. So these are the points that you have to write whenever a question is given on post-transcriptional modification. Not just write just about mRNA. Remember, you mention about tRNA and rRNA also, so that it will be useful. Right? So, okay. Right. Okay. Then, so any, this was one question you had, and the other one you said it is related to the techniques, right? PCR, you were saying. So we'll just look at PCR technique here. So let me just tell you, because I don't know where. Okay. So, right. So PCR, what you're supposed to write, you all know it is a polymerase chain reaction. So, so in this, why do you do this? We do this when we take a DNA of interest and we want to make multiple copies. That is why we do PCR. So first mention who was it who, the one who invented it. So you mention his name, Carey Lewis. This is the one who invented it. And then you start talking about the different steps in that. So what are the 3 steps in this? So you initially have the denaturation. After denaturation, we have annealing. And after annealing, we have elongation. Okay, or you can say polymerization, elongation, etc. So these 3 steps. So what are the indications for PCR? Now, PCR indication is, why do we need PCR actually? When we want a particular gene of interest, we have a doubt whether there is a mutation which is going on there. Or we want to know whether there is any viral particle which is there. Or we want to know whether any bacterium is there. So clinical diagnosis, like for example, we have TB, etc. Supposing the sputum of that person, you are not having so many bacteria. You are just probably having about 1 or 2 that you are not able to find out generally if you are doing a smear and seeing it. Then what will you do in that case? You will have to use this PCR. So this PCR is of varieties, but just remember, at least you have an normal PCR is one. The second one is called an RT-PCR, that is reverse transcriptase PCR. Okay, so this consists of what are the different stages. We said denaturation. So when denaturation has to take place, so you are going to cause separation of the two strands. So when you separate the two strands, you require a very high temperature. What is the reason? Because the bonds are so strong, right? We have double bonds, triple bonds between the nucleotide bases. So we have to cut that so that it becomes separate. So when it has to separate itself from that, you need a very high energy. It can go even up to 90 degrees centigrade. So such high energy, you will have denaturation. Bring the temperature again down. You bring it to probably around 50. So when you bring it down, then you will have annealing. How? With the help of what? With the help of primers. If you remember, in replication also you had a primer, right? And that primer was produced by what? By a primase enzyme. It was an RNA primer, right? But here, this primer is an oligonucleotide primer. Okay, so you have this primer which is complementary to the strand, that is to the ends of the strand of interest. So you might have a particular gene of interest. On the either side of the gene of interest, you will have a sequence which is complementary to this primer. Okay, you will keep that as a complementary to that primer. So once you have annealing happening, now annealing means it will join and get attached to that. So once that annealing happens, then finally, now you can again raise the temperature to around 70 degrees centigrade. So when you raise the temperature slightly to 70 degrees centigrade, this you have added into that test tube where you have all these. You have also added the deoxyribonucleotides you have added the DNA polymerase also you have added. So when you add the DNA polymerase, it will use this and it will start forming the new, that is a complementary strand. It will form in the 5' to 3' direction on both the strands of the DNA. It will form a complementary strand. Then, so now what has happened? These two strands have become 4. Then the 4 strands will become even more. That is doubling of that is going to happen. So this is how it will keep on. So 2 to the power of n, try to remember. So if it is going to be one strand, it's going to become 2. 2 strands are going to become 4. 4 are going to become 16. 16 is going to become more. So like that, it will keep on increasing. So it is always doubling will happen. And at the end of, say, probably 25 cycles like that, you will have more than 1 million copies will be there. So you will get your gene of interest. So you will be able to find out whether it is a viral particle, a bacterial particle, etc. You can find out. So that is about PCR. So this PCR you can other types of PCR if they ask you. The clinical importance I've already told you. Other types of PCR if they have to ask you, just mention about RT-PCR, which is reverse transcriptase PCR. So this we have seen that it has come in this COVID era. So what happened in the COVID era? We know that this the COVID virus, it is an RNA virus. So if you want to get the fragment and you want to actually produce more copies of that, you have to first convert it into a DNA, right? So you are going to use a reverse transcriptase enzyme, make this RNA into a DNA. Then you're going to find out a complementary sequence to that with using complementary primers. That is how you're going to do. So reverse transcriptase PCR is one. And after that, you can also mention about qPCR. qPCR means quantitative PCR. That it will tell you exactly how much of the content of the gene of interest you are having. It is also called the real-time PCR because it will tell you, okay, after 1 cycle, you're having so much of the gene of interest. After 2 cycles, you're having so much of the gene of interest. This is how it is going to tell you. That is why it is called the reverse transcriptase PCR. Okay. So with that, I hope I think I have reverse transcriptase. Any other doubts that you have? Okay.
Here is another question: is electrophoresis and blotting techniques are important topics?
Electrophoresis and blotting techniques are blotting is important, right? Blotting techniques are important. Electrophoresis, they did not ask you because they have too many questions to ask. They will not ask electrophoresis. So you can ignore electrophoresis. You need not study about that. But blotting techniques, you have to know. And I think it is easy to remember, right? Snowdrop is what the way by which you can remember. So snowdrop is, you can write SNOW, and then on the right side you write DROP. So S stands for southern blotting. So southern blotting is for DNA, right? Northern blotting, this is for RNA. The O's, you can ignore it. Then you have the W is western blot, and for that it is protein. You also have, there is one more blot, some call it dot blot, etc. So you need not write about those, at least write these three. Okay, southern blotting, northern blotting, and western blotting. This is important for you all, the blotting techniques. So for blotting techniques, again, you mention what is the use of a blotting technique. So the importance of blotting techniques, explain the blotting techniques in detail, write the clinical indications. So what are the clinical indications? Whenever a gene of interest you want to identify, you want to find out RFLPs, restriction fragment length polymorphisms, then you will use that. Or you want, supposing an RNA you want to find out. So that is any RNA of interest you want to do, you want to multiply that, then you can use this blotting technique. That is the northern blotting. Protein of interest you want to produce, or you want to be able to identify the protein of interest. So for the protein of interest, you will see that it will bind to its own antibody, and that is how you will be able to recognize it. So you just have to remember, blotting means they usually use a blotting paper. So that blotting paper will is the one which is going that this is the one where the DNA or the RNA are going to get fixed onto that. It will not be like an ordinary paper, but it will be like a membrane. It will be on which you are going to blot your DNA or RNA to it, or you can have your protein can be blotted onto it. Okay. So yeah, PCR, yes. Okay. Any other questions? Blotting, yeah, blotting is the one which is important, right? Okay. So you all are clear about translation, it looks like, because and replication, because nobody has asked about replication and translation. So if you get a question on replication, remember to draw the fork, okay, replication fork. So draw it, label it very well. Just like every answer, write the definition, draw the replication fork, label it well, explain in detail, initiation, elongation, termination, write a line about the inhibitors. So if you do it in this particular way, sure to get this. Almost they might give you 8 to 9 marks, they will easily give you. So it is in this form that you are supposed to write. So, okay, there is only one person. Okay, 9 people are there. Any other questions from the attendees?
There is another question. Ma'am,
Yeah.
in cancer patients, the telomerase activity is reduced.
In cancer patients, telomerase activity is increased, not reduced. See, telomere, they are present at the ends of chromosomes, right? That two eukaryotic chromosomes, which are linear. So the enzymes which help in lengthening the telomeres are called telomerases. So why should it lengthen? Because we know that after every replication, now this telomere is becoming shorter and shorter. Why is it becoming shorter? We know that you can add any nucleotides only from the 5' to 3' direction. You can't add nucleotides from the 3' to 5' end. So what is happening? So that is why we have Okazaki fragments. Finally, at the 5' end, the Okazaki fragment will be removed. But we require a primer to actually keep on adding some more sequences, which it cannot add. So therefore, what happens is at the end of the 5' end of the DNA strand is going to become shorter. So when it keeps happening like that, shorter and shorter, the telomeres are lost, then it's not a problem. But if it continuously happens, it will start even damaging the exons, which are there very close to the telomeres. If it damages the exon, some proteins may not be synthesized. So that is why we should be careful, and the body tries to lengthen it. So lengthening, where will it happen? It happens in the stem cells. It will happen. So usually it happens in the stem cells where it is continuously, these telomerases are active. But in cancer, what happens? That cancer cell has telomerase activity, which is very high. So it keeps on multiplying, it keeps on producing a lot of proteins, etc. These cells, that is why they are called immortal cells, right? They continuously multiply. Their growth factor production is very high. So what now scientists are doing is they are developing drugs to inhibit the telomerase activity. So when it is inhibited, the telomerase activity, then it can cause the death of the cancer cells. So that is what you have to know about telomerase, right? Okay. Then can you explain replication? Huh? Okay. So replication, let us just look at replication. Replication may come for you all as an essay question. See, definitely it usually comes as an essay question only, replication. Sometimes they ask about translation as an essay question. Okay. So let us just look at some important points in replication. So let me share the screen with you and try to explain, right? Okay. Right. So replication, you have to write about the definition of replication, and you have to mention that it takes place in the S phase of the synthetic phase of the cell cycle, right? This is how you write first. What are the characteristic features of replication? So the characteristic features of replication are, it is semi-conservative. You all know the meaning, I hope. So definitely in the daughter cells, you have a parent strand and a daughter strand. So half the original DNA strand is saved and it is present or conserved in the daughter cell. Then it starts at the origin of replication. Synthesis is always in the 5' to 3' direction. That is a compulsory thing. Synthesis is bidirectional. That is, you have an origin of replication and it can move to the left or to the right. It is bidirectional. It is semi-discontinuous. You all know that when you have two strands, on one strand, what are you having? Continuous synthesis. On the other strand, you have discontinuous synthesis. So that is why it is called semi-discontinuous. And we know that initially RNA primer is required to start the replication process. So this is just the same thing is being explained in this. Now, what are the steps in DNA replication? So the steps in DNA replication are, first, you have a protein which will identify the origin of replication. Origin of replication is called ORI, O-R-I, ORI. So it is recognizes that. Once it recognizes that, then this protein which is recognizing this region is rich in 80 nucleotides, A and T, adenine and thymine. So these nucleotides, it is rich in that. Why it prefers AT and not GC? Because AT sequence is going to have double bonds. GC will have triple. To break that double bond is easier compared to using energy to break a triple bond. So that's why it will cause unwinding of this double strand. So it starts there and slowly starts separating. When it separates, what is forming? A replication bubble starts forming. Then as this bubble increases, now you have enzymes which are called helicases will come into play and they'll start separating the strand. If it is a eukaryote, you have to talk here again about acetylation of histones, which is separating itself from the DNA, and then you talk about the origin of replication. So the three major steps are these: initiation, elongation, and termination. And in initiation, we spoke about this is a picture of a prokaryote where you have a single replication bubble, and then for a eukaryote you can see you have multiple replication bubbles. So this will be the origin of replication. That's why you can see on this strand it is going this way, on this strand it is going the other way, right? The origin of replication is here. And whenever we have a replication fork, this is the center, so you have a replication fork is this, right? So now let us look at what happens in RNA replication. So what did we say now? Helicase is coming. It is an enzyme which is separating the two strands. Now when the two strands are separated, they would like to again join back. So to prevent that, what do we have? We have single-stranded DNA binding proteins which are binding to that strand, and they keep it separately. Then you have an enzyme which is called a primase. Now, when this primase is there, we are talking about in prokaryotes. In eukaryotes, we have an enzyme which is called DNA polymerase alpha. Now, that has a primase activity also. So what is the function of a primase? Primase, what it will do is it will add ribonucleotides, it will add. So you can see here, it has added some ribonucleotides. So this is the template strand which is in the 3', 5' direction. You are going to have, it is adding the RNA primer here. This is the function of the primase. Then after it has added a few nucleotides, now DNA polymerase 3. Now this DNA polymerase 3 means it is a prokaryote. So this prokaryote will, the enzyme DNA polymerase 3, it will continuously now start adding. So depending upon the strand, you are having a leading strand, it will do continuously, it will produce. Lagging strand, it will produce discontinuously. Okay. Now what is required for that? What is required here is ATP is required, or whatever is the nucleotide, a trinucleotide rich phosphate is required for that. So it will take that nucleotide triphosphate or deoxyribonucleotide triphosphate will take, and from there it will remove the pyrophosphate part, and only it will attach the AMP will be attached to it. Then the bond, you know that you are going to have a phosphodiester bond being formed. So the phosphodiester bond will be formed with the energy which is released by breaking down the pyrophosphate. So PPI is going to break down to form two inorganic phosphates. That energy will be used in forming the phosphodiester bond. And it always, DNA polymerase sees to it that the correct bases are going to get attached. That is, if it is going to be A on the template strand, it will be T will be there on the daughter strand which is being formed. So it will always check to see whether it is coming. If it is G, then it will have C. So this is how it keeps on attaching itself. This is DNA polymerase 3. So so here you can see this is the helicase which is trying to separate the two strands. These blue ones are all the single-stranded DNA binding proteins which are here. Okay, so that is what you can see. And this is what I was talking to you about. So now this helicase is moving like this. This is how it will move. So it is coming from here and it is continuously, it is going to move like this. So when it is moving this helicase, you can see that this is the helicase here. So it has separated the two strands, and you can see the single-stranded DNA binding proteins. You can see it here. Then you also have a primase enzyme. What is this primase enzyme doing? It has added an RNA primer here. After it has added an RNA primer, the DNA polymerase 3 is going to attach a lot of nucleotides one after the other, and it is going to go continuously. Therefore, this is called the leading strand. What will happen in the other side? You can see bits and pieces of primer are there. Bits and pieces of the DNA fragments are there. So this is called the Okazaki fragments. This is called. And how are they added? You can see it is going in an opposite direction. It is going away from the replication fork. Okay, so it is because it has to add only in the 5' to 3' direction. So you can see there is one RNA primer here, there is another RNA primer here, another RNA primer. To do this, it is difficult. That's why it can only do it in bits and pieces. So these are the Okazaki fragments. Then as it is separating the two strands, what is happening? Supercoils are being formed. Now these supercoils will be cut by these topoisomerases. So they will cut the supercoils and they will join them, so that it will again become a single strand, so that this helicase activity will continue without any problem. That is why it is done this way. Then after that, what is going to happen? After that, now all these primers have to be removed, right? Because they are still RNA. So this has to be removed with the help of an enzyme which is called DNA polymerase 1. So that will degrade and completely remove these primers. After it removes, you have a gap. So that space which is there, again it is filled by the action of DNA polymerase 1. So what it will do, it will keep on adding deoxyribonucleotides now, so that it will fill that gap. And the last, between the 5' end and the 3', 3' hydroxyl and the 5' hydroxyl, there will be one small gap which is there. That gap is going to be sealed by a ligase enzyme. So this much, if you draw this diagram and you show this, your entire replication will come here. You can finish that. Just remember all the important enzymes here, right? Then you have to write about the functions of DNA polymerase 3. So DNA polymerase 3's main function is what? It has a 5' to 3' polymerase activity. It also has a 3' to 5' exonuclease activity. What is the function of the exonuclease activity? Whenever a wrong pairing happens, it will remove that abnormal nucleotide and add the right nucleotide. So it has two activities, right? DNA polymerase 3 has two activities. What are those two activities? One is a 5' to 3' polymerase activity. It also has a 3' to 5' exonuclease activity, right? Then what about DNA polymerase 1? DNA polymerase 1 has three functions. What are those three functions? It has not only a polymerase activity from 5' to 3', it has an exonuclease activity, 5' to 3' and 3' to 5'. So that is why, because this 5' to 3' activity is there, it is able to remove the entire primer. All the primers are removed by that. And because it has a polymerase activity of 5' to 3', it is able to add new nucleotides. Okay, so that it is going to do. What about DNA polymerase 2? It has only repair activity. So prokaryotes, very easy. Remember these three enzymes. DNA polymerase 3, main enzyme. DNA polymerase 1 has a lot of activity, which is very important to remove the primers, to fill the gaps. That is why it is there. Then we have finally the third one, which is DNA polymerase 2, whose function is only repair. And you have to remember these proteins. So we know the action of topoisomerases. Whenever supercoiling will happen, it will see to it that it will cut those supercoils, unwind it, and again ligate it, so that the strand is fine, there is no supercoiling, and the helicase can proceed without any problem. Helicases, just like a zip opener, that's what they give. So it separates the two strands. What is the function of a primase? For RNA primer synthesis. Single-stranded DNA binding proteins to keep the strand separately. DNA polymerase, synthesis of the new strand. DNA ligase, the gap between a 5' phosphate and a 3' hydroxyl to join that, seals that nick via the phosphodiester linkage. Okay, so that is about this. And one more thing, the termination. This is in prokaryotes. You have two sites. One is called the termination site. You have a TUS protein. If the TUS protein is coming and recognizing the termination site, the prokaryote, it will stop. Okay, eukaryotes, still they have not found out exactly how it stops, but it stops. Now, mammalian polymerases, you have to know, that is eukaryotic polymerases. You can see alpha, beta, gamma, delta, epsilon. So it is easy to remember these. And remember that this polymerase alpha also has primase activity. That much you have to remember. Polymerase beta, over there it was DNA polymerase 2 in prokaryotes, which had repair activity. Polymerase beta has repair activity. Gamma is mainly for mitochondrial DNA. Polymerase delta is for leading and lagging strand synthesis, also for proofreading activity. And polymerase epsilon for leading strand synthesis and proofreading activity. Just mention the differences, that's all. Then you can also, if it is easy for you to remember, you can also mention the differences between prokaryotes and eukaryotes. So I'm not going to explain this in detail. When you look at the video, you will understand that this, if they ask you as a question, you can write this differences between prokaryotes and eukaryotes. There are minor differences. Like, for example, single-stranded DNA binding protein in prokaryotes, whereas in eukaryotes, you call it replication protein A. Here, you are going to have something called a sliding clamp, which is a part of DNA polymerase 3. Whereas for eukaryotes, it is called pCNA. So what do we mean by a sliding clamp? if you imagine this is my finger, which you can see is the template strand. Now you can have the DNA polymerase. It has to now hold on to this properly, right? So the clamp is a patakara. So it has to now fix itself onto that. So if it fixes onto that, so the DNA polymerase will actually, in a prokaryote, will fix itself onto the DNA's template strand. Whereas in eukaryotes, you have pCNA, which is acting like a sliding clamp. Then what is that which is loading the clamp? You are having a protein which is called replication factor C. Whereas for a prokaryotes, it is the same. The clamp loader is the same. The sliding clamp is also the same. Whereas here it is different. Then you have to learn about the inhibitors. Prokaryote, these are the inhibitors. Eukaryotic replication, these are the inhibitors. Telomerases are not there in prokaryotes. In eukaryotes, you have the telomerases. So this is in brief about DNA replication. Okay. I hope that answers your question. Right. So with that, I think we have completed. So any further doubts? Is it Naimisha? Is it over?
Yes, ma'am.
Yeah. Right.
Let's
Yeah.
end చేసేద్దాము.
End చేసేద్దాం.
Let's end the session with the Shanti mantra. Sarve bhavantu sukhinah, sarve santu niramaya, sarve bhadrani pashyantu, ma kaschid dukkha bhavet. Shanti, shanti, shanti. Extend our heartfelt gratitude to Dr. Vijaylakshmi madam for an insightful and enriching session, madam. Thank you, madam, for your valuable time and guidance. We also thank all the participants for your presence and active participation. Tomorrow there is a session on the topic about vitamins and coenzymes. The topic is taken by Dr. Kirana madam, Professor of Biochemistry, SWIMS. We look forward to your active participation in the upcoming sessions of the Rapid Revision series. Thank you, madam. Thank you.
Thank you very much. Thank you, attendees, for participating, and thank you, NMO. Thank you.